So good morning, everybody. My name is Ellen Harrell. I'm the Executive Director for the Louisiana Grazing Lands Conservation Initiative. And thank you for joining us for webinar 2 of our Winter Forages series. Today we have Dr. Lisa Fultz with LSU speaking to us about what is actually going on underground and giving us a scientific perspective of soil health. So we're really excited to have her here with us. She's going to have a brief presentation and then we'll have an opportunity for question and answer. But as we're going through, and if something's really jumping into you, feel free to put it in the chat or just write it down so that you don't forget. Or she might pause to ask if you have specific questions. But we really appreciate y'all being here with us today. And be sure before the end, we will do a t-shirt drawing. So you guys are eligible for a t-shirt by being on the live webinar, and that's what our t-shirt looks like. So really excited about that. But Dr. Fultz, thank you so much. And if you want to get started, we will just let others in as they log on.
Thank you very much. And I want to thank Ellen and Tara for inviting me to do this, and everyone for being here this morning. I know it's pretty cold outside, so hopefully everybody's staying warm. And so as Ellen was saying, my name is Lisa Fultz. I'm the Associate Professor for Soil Microbiology at LSU and with the LSU Ag Center. And a lot of my research focuses on soil conservation, soil health, and what different management practices we implement and how they influence those properties. I work in a variety of different management systems, from traditional row crop production, grazing land systems. This is a project I'm working on with somebody at Southern University, looking at pasture land systems, to looking at water quality issues and how what we do affects water quality as well. So we'll go ahead and get started. If my computer will cooperate, maybe. There we go. So one of the first questions is, so what is going on down there? And we have a fairly decent idea, I would say, of what kind of processes are happening in the soil profile and how the soil biological community is contributing to that. And here what you see is a diagram with soil biota in the middle, and just looking at all of the different properties in the soil that and the functions of the soil that are related to how the soil processes different things and how those relate to soil health. So we know that soil biota can help with improving aggregate formation and aggregate stability, which in turn benefits and improves things like water infiltration and water storage in the soil profile. We have all these organisms. You can see the earthworm here in the background. I get really excited when I see those in our fields because it means that we are creating habitat that is hospitable for those organisms. They're helping to break down that organic residue, which is in turn aiding things like nutrient cycling and detoxifying things like pollutants in the soil profile. As they break that material down, they're also releasing gases like CO2, nitrous oxide, and methane. So they're influencing atmospheric compositions. They can help with plant protection and pathogen suppression. So they really do fulfill a lot of different roles. And one that we'll go into a little more detail in a little bit, though, is this process of nitrogen fixation, which is an ability of specific bacteria in the soil that can actually take nitrogen out of the atmosphere and convert it into a form that is used by plants in plant available forms. So just to get us kind of started, I want us to be on just some basic soil properties that all influence plant growth, but also influence the habitat that these organisms are found in. And one of the basic ones that we talk about quite a bit is soil texture, looking at the distribution of sand, silt, and clay that is going to determine things like cation exchange capacity and nutrient retention in the soil. It also influences things like pore space, and those pores are where things like roots and microbes and macro and microfauna are living in the soil profile, and water retention and water infiltration. So it really does also play a role in how our soils function. Things like soil pH, which, as we know, influences nutrient availability. And you can see here in the top corner, we know that several of the plant essential nutrients are more readily available in sort of that 6 to 7 pH range. but it also influences things like the microbial community. You can see fungi have a pretty stable bar all the way across. They're not as influenced or impacted by soil pHs, but bacteria are less well adapted to those low pHs. And so we tend to see bacterial communities start to decline as we get into more acidic conditions. We also see this property of cation exchange capacity, which is essentially a function of the soil having a negative charge on the soil surface. And that negative charge allows for the soil to hold on to nutrients like ammonia, calcium, potassium, phosphorus in the soil profile. And that is influenced by things like soil texture, but it's also greatly impacted by soil organic matter. And soil organic matter is
a lot of grass.
Is really influenced by or has a very large influence on the microbial community and what's going on in the soil profile. Organic matter is simply all of the decayed and decaying plant residue, microbial community, exudates that are coming off of root tissue that are part of that. It also includes things like the microbial biomass, which is on average somewhere around 2 to 5% of that organic material. And you've probably seen with a lot of talk about carbon sequestration, soil organic carbon, which is a component of that soil organic matter. And this is really the sort of nutrient source and the carbon source for the microbiota that live in the soil. So bacteria, fungi, nematodes, all of those are living off of this organic material. And of those, remember I said that sand, silt, and clay will influence cation exchange capacity. Soil organic matter actually has a much larger impact because soil organic matter is a very diverse structure to it. And so it can actually increase cation exchange capacity by a very large amount, which can increase nutrient retention and water retention in your soil. It provides protection and habitat for the microbial community and increases water retention in that soil profile. And I'm going to show some data later that looks at the relationship between things like increasing soil organic matter and how that influences the microbial community, nutrient retention, and nutrient turnover, as well as improving aggregate stability. So this is looking at a stylized diagram of what's kind of going on in the soil. You can see we have our sand, silt, and clay, so our big pieces of sand, and then our silt and clay and our smaller components here in the profile. We see a plant root growing through there with all of its lateral and smaller roots coming off. Saprophytic fungi is fungi that's feeding and living off of dead and decaying material. So that's here wrapped around this root, collecting exudates that are coming off and using the byproducts of bacterial degradation, that kind of thing. We have mycorrhizal fungi, which actually are interacting with the root, and they are providing access to nutrients like phosphorus, calcium, also access to soil moisture. In exchange for that, they will give those nutrients to the plant root for carbohydrates and sugars from that material. So they create the symbiotic relationship where they're trading resources between them to persist in the soil. We see those pore spaces with the water films, where we have things like nematodes and protozoa moving around through those and you accessing organic matter and exudates. We have those open air-filled pores where things like mites and earthworms will live. And then we have these smaller pores here, and you can see one that's kind of blown up where we find our organic matter, our bacteria, and those kinds of smaller organisms that are collecting in those areas. And so this is sort of the idealized version of what the soil looks like. Something to keep in mind is when we look at sort of average soil conditions with a bulk density that is around 1.3, about 50% of your ground or your soil that you're looking at is actually pore space. Only 50% of that is solid material. And so we are very dependent on that sort of even distribution to provide habitat, not just for the microbes, but also plant roots and the growth of those organisms. So what happens if we disturb that system? Tillage is a very common practice and does have potential benefits that come with it, and that's why it's been utilized for so long. It does allow for seed bed preparation, which allows for you to have good seed to soil contact. So you're getting good germination of whatever you're happen to be planting into that soil. It's been used for weed control in particular. It can help improve soil aeration and ameliorate compaction issues that you might have, and also help with amendment incorporation. So there are some benefits that come with tillage, but we also know that for all of those benefits, there are costs that are associated with the utilization of tillage. It is disrupting that soil structure. So if we go back and we look at that sort of idealized version, we are breaking up and those aggregates and that organic material that's there. We're breaking up fungal communities and fungal mycelia. Reducing pore space, which is going to restrict movement of water, roots, and microorganisms through the soil profile. We also see a loss of that surface residue as it gets incorporated into the soil profile. And this can be an issue, particularly in areas where we see high intensity rainfall events. I don't know how well it shows up, but the picture up in the top corner is looking at a rainfall as it impacts the soil surface. And you can see that just the energy behind that rainfall that it imparts into the soil, and it pushes those particles away from each other. And if you look at the soil and you think about all of the amendments that you've added to it, the nutrients, chemicals, the organic material that you've incorporated into that soil, as that material runs off, that's a resource that you're losing from that soil profile. So when we do expose those soil surfaces, we see an increased susceptibility to soil erosion throughout that soil profile. And so for those reasons, tillage may serve a very important purpose, but we are looking at ways to reduce the frequency of tillage that's used, or maybe the disruption that comes from that, looking at processes like strip till or shallow tillage, where you're getting some of those benefits with the incorporation of the root residue or seed bed preparation, but minimizing the extent of that disturbance. But what happens if you decide to convert from a till practice to a no-till practice? Many people are probably familiar with, and one of the reasons that a lot of people are nervous about adopting no-till is we do have a tendency to see a decline in yield immediately after adopting no-till practices. And that comes from this transition of going from a biological system that is used to seeing that incorporation over a very short time. They break down that organic material to one where they're having those roots and biomass influencing the system over a long period of time. So we do see a restoration of nutrients into that profile and nutrient retention, because we are seeing an increase in soil organic matter. With that soil organic matter, we're also seeing increased cation exchange capacity. But in addition to that, we're seeing increased competition for resources like nitrogen. Because you're seeing that transition of the microbial community and they're utilizing the soil nutrients to help break down that organic material, they're going to get to the nitrogen before the plants do. And so they will use up and immobilize some of that nitrogen, reducing its availability. And that is going to contribute to that initial yield decline as the system sort of resets itself and finds a new balance. We'll also see a rebuilding of aggregate stability as we have those plant roots and the mycelia binding the soil particles together. And then we also see the release of organic glues, things like glomalin from fungal communities that help to hold the soil particles together. We see, because of the improved aggregate stability, increases in water conservation and crop water use efficiency, because they have that as an accessible resource. Fewer field operations and days needed for planting, which relates to reduced fuel usage, which everybody was looking for ways to reduce their fuel usage the last couple of years just because of how expensive it became. We see reduced losses due to soil erosion, because we are rebuilding that sort of soil health, those properties that help keep the soil in your field rather than running off into your local water bodies. One thing we also see is some stratification of soil nutrients, because now you are applying those soil nutrients typically at that soil surface. You also have that the plant roots and the leaves and tissue being applied there at the soil surface. As they build up, you don't have that mechanical mixing that helps incorporate that into the soil profile. So now you're reliant on your biological community. So things like earthworms, beetles, larger organisms like moles and mice to help incorporate that organic material down deeper into the soil profile, which takes time, particularly when you're used to the immediate response that tillage can give you of incorporating that material at a very quick pace. And then we have this thing of when we add the fertilizers to that system. Now, whether this is true, whether you're going for no-till or a tillage type system, a lot of that's going to be dependent on what kind of fertilizer are you adding. If you're adding a chemical fertilizer, something like urea, potash, UAN, you're doing that specifically to feed the plant. There's generally not a lot of excess material and nutrients that are going towards the microbial community. And with the exception of something like urea, which will generally go through a biological process to be converted to nitrate, that's material that's going to be used by primarily the plants and some of the microbes living in the soil profile. Now, when we apply it in the form of organic fertilizers, so things like compost, manures, something like that, those nutrients that are tied up in that material are not immediately available because they are part of that organic material. So they have to go through a process called mobilization, where they are converted from an organic form to an inorganic form that is available for plant uptake. So they get incorporated into that organic material. They start getting broken down by soil microorganisms, enzymes that they produce that help break that down. So we have enzymes that will help break down things like cellulose and chitin from the fungal communities, things that'll break down urea bonds and all of that in the soil profile. As that starts to break down, those nutrients then become incorporated into the soil, and they may become plant available, or they end up in other organisms as they get assimilated into their biomass, and we have to wait for something to eat them for those nutrients to become available. So it kind of depends on what your immediate needs are on sort of where those nutrients end up. Other things that are going to influence that, again, we talked about cation exchange capacity. So nutrients like phosphorus and potassium are going to sort of stick to the soil surface, whereas things like nitrate nitrogen, if you apply that, it is very mobile in the soil profile. So it will move through the soil profile every time you get an irrigation or a water event. And so you can, some of the decisions you make about when you want to put those out may be dependent on what type of fertilizer that you're using as well. And this is looking at what happens from a biological component of how those nutrients get turned over. So we start out here. Most people have probably seen some representation of the soil food web. This is actually a newer one that I came across the other day, and I thought did a very good job of showing this. You have your primary producer, which is your plant, and it's going to be taking in CO2 for photosynthesis, releasing oxygen into the atmosphere, and it's also going to be pulling in nutrients through its root profile and concentrating that in the plant stems, the leaves, and the root material. As those tend to grow, we see loss of root cells, we see exudates coming from the soil root profile, the leaves drop and everything, and that now becomes an organic source and a nutrient source for the microbial communities. So we see things like nematodes that feed off of roots. We see earthworms that will actually burrow up to the soil surface or live in that organic matter at the soil surface. They break that down and they excrete cast that have all of those nutrients, again, concentrated in that material. We also have bacteria and fungi that will live in the rhizosphere, which is that zone of soil that is adhering to and influenced by the root material. They're feeding off of the root exudates and things coming off of there, producing and converting some of those nutrients into plant available forms, and they're tying some of those nutrients up into their fungal cells, so their cell walls, their mycelia, and that kind of thing. And so then when we go into the secondary community, where things like arthropods, other nematodes that feed off of fungi and bacteria, protozoa that use those, they will eat and they will poop, because everybody poops. And those nutrients that are then tied up in those become a little bit more available, because none of these things are 100% efficient. If they take in 100 grams of carbon, they're going to use somewhere around 60% of that carbon. Then there's 40 grams of carbon that gets left over for conversion to CO2, organic material as a waste product, and that kind of thing. And that sort of happens at each step through that process. So there's different ways that can get incorporated into the soil profile. If we introduce something like animals to that system, cattle, goats, anything feeding off of that, they also are helping to cycle those nutrients as they use that. The animals will take it in, whatever they don't utilize for themselves, they'll defecate back in or urinate back onto the soil surface. And now you have a way for those nutrients to cycle that way as well. And so you can see how they're all interconnected to help with cycling those nutrients back and forth. And this kind of ties back into the fertilizer idea, because in general, if we're not taking anything off of that site, those nutrients should constantly cycle through those materials. It's when we harvest material, whether we're harvesting a cornfield or pulling cattle off of the field for to take for whatever uses we have, those nutrients, we start taking them off site, we have to replace them somehow. And that comes in the form of either organic amendments like manures and compost material, or chemical fertilizers as well. And so that's sort of how that all comes together. Another way that you can bring nutrients into the system and help keep nutrients there is something like a cover crop. And these are actually something I've spent a lot of time on. Some people actually call me the cover crop lady within the ag extension system, which is cool. At least they know who I am. But I do want to look at some of the different ones, because they do have some similarities between what benefits they can bring to the system. But they each have sort of a unique property to them that may make them beneficial to whatever needs that you have. So in the case of grasses, we're thinking of things like cereal rye, annual ryegrass, particularly in like grazing systems, bob or black oats, wheat, and they are extremely good at scavenging nutrients, particularly because they can put on some very deep root systems. And if they're allowed to grow for a long period of time, you can have, particularly in perennial grassland systems, roots that will go meters down into the soil profile. So not only are they scavenging the nutrients that maybe get applied here at the soil surface, but nutrients that maybe have moved down further into the soil profile that they can cycle back up to the root surface or the soil surface and make available for other organisms. And we've seen a very impressive weed suppression coming from using grasses, in particular some of our row crop fields where we have invasive Italian ryegrass. Using things like cereal rye pretty much knock that material out. Brassicas will do some of the same processes. They do help with scavenging nutrients and breaking up soil compaction. But something that they sort of bring to the mix that the others do not have the ability is that they produce this compound called glucosinolate. And as that particular compound breaks down, one of its byproducts is an isothiocyanate, which is known for being a fungicide and an nematicide. So if you're in a field where potentially you have an issue with nematodes or fungi causing issues and diseases in your plants, you might be able to look at incorporating some of these brassica materials, particularly things like radishes, and to help with those issues. And then the last group is our legumes. And legumes have this amazing ability to make a symbiotic relationship with bacteria in the soil. In particular, we're looking at things like rhizobia, frankia, that will utilize an enzyme called nitrogenase. This nitrogenase enzyme helps to take nitrogen from the atmosphere and convert it into that ammonia compound that they then share with the plant. And that what happens, you can kind of see it here, the plant will demonstrate nitrogen deficiencies. This will cause the roots to send out signals into the soil that indicate to the rhizobia that they're needed. As they infect the plant roots, the plants will actually create protective coatings or the nodules in those systems to protect them. And that helps also protect the nitrogenase enzyme, because the nitrogenase enzyme will actually break down in the presence of oxygen. So they create these habitats for them. And in exchange for that, the rhizobia will fix nitrogen and provide it to the plant, and the plant will give them carbohydrates and sugars, and they create the symbiotic relationship. What they've also found is that phosphorus is potentially a limiting factor in this relationship, because they need that phosphorus for root elongation and root growth. And so they're starting to see some of these, or identify relationships between not just the rhizobia bacteria, but fungi like arbuscular mycorrhizal fungi, which increase our access to plant available phosphorus. And so there's actually additional signaling that's happening saying we need more phosphorus, and they will make that relationship and start sharing phosphorus between the resources as well. The important thing to keep in mind with this is that if you have ample available nitrogen in your soil, there's no need for nitrogen fixation to happen. And so your plants might create nodules, but if you break that nodule open, it may be sort of at the same tannish color as the surrounding root system. And what that means is there's no nitrogen being fixed. Now, if you break that nodule open and it's a pinkish red color, that means that that bacteria is active and it's actively fixing nitrogen. And so if you don't see that color, you might look at your soil test data and see, do we have excess nitrogen present? Is there something that may be deactivating that relationship? And so I just want to take a few minutes, because I know I'm probably going over a little bit, and look at some of the data that sort of relates all of these things back to each other. So some of this is coming from some trials we've had. The data I'm going to show is at 5 years into the study, but we've actually had land in no-till and cover crops for about 9 years at this point. It was started just before I came to LSU, and we've been maintaining it. And you can see we have our rows. This is in the very early part of the project. This is planting into our cover crops into our corn residue. You can see all that material there on the soil surface. And this is some of our cereal rye. It's coming up in the different plots, and we get a really nice stand of material here. And we've actually been able to demonstrate that when we utilize those legumes that are bringing nitrogen into our system, we actually can get a nitrogen credit from that material and potentially apply less nitrogen to our system than what we would do in sort of our standard practices. So the first one I want to look at is, this is a soil enzyme, actually 2 different soil enzymes. We have one enzyme that is responsible for breaking up cellulose, and that's these first sort of orangish bars. And this is looking at over time, the buildup of that enzyme in the soil. And you can see that in the first 5 years, we had increased that enzyme activity, and so breaking down that material by 84%. So that's a pretty significant improvement in the formation of that enzyme in the soil profile. The second enzyme in this sort of greenish colored bars is one that is specific to nitrogen turnover and nitrogen cycling. And that enzyme has actually increased by 119%. So we're seeing the response of the microbial community that are producing these enzymes to help break down and incorporate that organic material into the soil and release those nutrients into the soil profile for use by the plants and other microorganisms. This is a study that I did looking at integrated crop livestock systems. And just to show you sort of the relationships between all of these, we had systems that they were sampled in 1997 before we put them in, sampled again 13 years later. And you can see we increased soil organic matter in our integrated crop livestock system by 31%. That may not seem very impressive, but this is in a system where they got maybe 18 inches of rainfall a year. And so seeing that kind of benefit and improvement in soil organic matter in a relatively short amount of time was actually pretty exciting for us. We also looked at systems that had been in place for about 5 years at that time, and compared and looked at them in relation to farmer practices. So this bar on the end is actually a producer that had Old World Blue Stem for part of his field, and the other part was in row crop. And when we compared that to continuous cotton production in West Texas, we saw a 50% increase in soil organic matter and soil organic carbon. So we are seeing the benefits of sort of adopting some of these conservation practices. And the reason that that's important is that when we look at the relationships and the microbial communities in some of these samples, what we found is that when we look at something like fungal diversity, in those monoculture cotton systems, they're fairly low, about 400% or 400 compared to in our rotation and perennial systems and our integrated crop livestock systems, somewhere around 12 to 1600, looking at those diversity measurements. And so this is actually looking at fungal communities across those. So again, we see those benefits, and those increases in organic material are being related to increases in fungal diversity and fungal communities. And then again, when we look at increasing fungal diversity, we have our monoculture cotton systems and our integrated systems. You can see that we're also seeing an increase in soil organic matter. And remember, soil organic matter is where we get things like cation exchange capacity and nutrients available to microbial organisms. This is looking at specifically arbuscular mycorrhizal fungi, and these are the fungi that will make associations with like 95% of the plants on the planet to exchange nutrients for carbohydrates. And we can see this is a measurement of aggregate stability. So higher mean weight diameter is better aggregate stability. And as we see the population of arbuscular mycorrhizal fungi increase, we also see improvements in aggregate stability. So kind of just going back to that initial diagram that I was showing you, where we have these relationships between all of these different soil properties. This is looking at the relationship between soil organic matter in the same fields and aggregate stability. And what was really interesting here is that all of these points are our annual fields. And you can see there's not a big difference. While we do see improvements in organic matter, we're not seeing an improvement in the aggregate stability, because these were systems that were routinely tilled and experienced disturbance at least a couple times each year. Whereas when we got into our perennial systems, where we had our grasslands, anything from Bermuda grass to overall bluestem, we can see the aggregate stability is increasing, and we also see higher than 2% organic matter in these soils. This is looking at data here in Louisiana at a producer. Actually, technically, this is in Mississippi. This is a producer who's about a mile over the Mississippi-Louisiana border. And here I just want to show that we see an increase in soil organic matter across his fields. And that is related to increase in total microbial biomass, so just the overall amount of organisms that are present. And then as we see an increase in total microbial biomass, we're also seeing increases in plant available nitrogen and plant available phosphorus. So again, seeing that by increasing these properties, we're seeing greater nutrient cycling throughout that soil profile. And then this is looking at a specific enzyme related to nitrogen release and nitrogen turnover. This isn't particularly important because one thing we saw in his fields is that inorganic nitrogen, so nitrate nitrogen and ammonia nitrogen, were actually decreasing over time, which would make you think that, well, we were seeing decreases in yields, but he was actually not needing to put out any nitrogen fertilizer in these fields because his total nitrogen, so organic and inorganic nitrogen, was actually increasing slightly over time. And so what was happening is when those microorganisms were active, they were utilizing things like this nagase enzyme, and helping to take some of that organic nitrogen and convert it into an inorganic form that the plants could use. So even though he wasn't having to put out any nitrogen fertilizer, his system was supplying the nitrogen that it needed for continued grass growth for grazing in his systems. So just to kind of bring it back to the initial diagram and look at how the soil biological community influences a number of different soil functions, and also to keep in mind that these different soil functions, so things like aggregate formation and residue decomposition, also interact with each other and feed different soil properties as well. And just to end it, and a little plug for myself, we are hosting a cover crop conference in February, February 14th and 15th here in Baton Rouge. This is the third one that the Southern Cover Crop Council has done. And if anyone is interested in cover crops, I invite you. There's the registration information, and I will open it up to questions. I see. Sorry, I know I started going a little fast there at the end.
Lisa, that was some great, fascinating information that I certainly didn't know all those details, and I'm sure that we're all going to have to go back and rewatch this and really take it all in. I came up with a few questions, and number one, that's great to know about the conference. We will put that on our master calendar that we've been compiling.
I can send you the
Um,
flyer if you'd like.
That would be wonderful.
Okay.
Kind of a question that I came along with.
Mhm.
And I'm pretty sure I'm familiar with, I think a lot of us are familiar with the producer that you're talking about that's just above the border.
Yeah.
How long have they not been using fertilizer
So
now?
at the time that we had done this, he had been doing it for about 9 years. And I believe he had not been adding nitrogen fertilizer for most of that period. And what he was doing was once his summer grasses had started to die off, he was planting a 9-way cover crop mix. I forgot to mention that included things like legumes and a few other winter grasses that he was using for grazing throughout that system. He was also incorporating rotational grazing. He goes out very frequently and moves his cattle so that they're moving through that system and helping to cycle those nutrients pretty regularly.
Yes, that's awesome. So, as far as that, let's see what questions I had. That length of time that it takes to convert, you were talking about the organic fertilizers are slower because of mobilization. How, number one, what are some examples of organic fertilizers? And number two, like how long before planting ryegrass per se, since we're talking about winter forages in this, how long before planting should you apply an organic fertilizer?
So organic fertilizers can be anything from a compost to manure that you get from whatever source. I'm more familiar with well, actually, most of my master's was working with animal waste. So things like poultry litter from feedlots, the beef manure. You could get swine. Well, I don't actually know if you can get it. We used a lot of swine effluent through irrigation systems. Any of those would count as sort of an organic source of fertilizer. Also the biomass, if you're using a cover crop, would consider it's basically anything that the soil is going or the microbes are going to have to break down to get those nutrients into a plant available form. That also comes with things like carbon as part of that process to help break it down. So that's sort of how it separates itself from chemical fertilizers. A lot of times they have some filler material, but you're primarily interested in something like nitrogen, phosphorus, potassium, or sulfur. And so there's not necessarily anything that's also feeding the microbial community in addition to providing those nutrients. Now and the timing for release is very important. I know for cover crops, what we saw is that one of my students looked at the changes in soil nitrogen after termination. And as far as nitrogen was concerned, we saw increases in soil nitrogen somewhere around 4 to 6 weeks after termination. So you'll start to see the benefit of that nitrogen relatively quickly. But the thing to keep in mind is when you apply that organic fertilizer, you're not getting all of that. So you're going to get somewhere between 30 to maybe 50% of that nitrogen or whatever you're applying, depending on the source of material that you actually apply. So you kind of have to keep that in mind when you're doing your application rates, is you're not going to see all of that. The other thing to keep in mind with organic fertilizers is that when you apply for nitrogen, you're going to get potentially high amounts also of things like phosphorus and potassium, which may be beneficial. But if you have soils that are already very high in phosphorus, you're over applying what you need, and you might see this buildup, which doesn't necessarily sound like it's a bad thing. But if that phosphorus runs off into local waterways, then you have a problem with algal blooms and that kind of thing. And that's a situation that Arkansas actually got into because they had producers with poultry houses that were just continuously putting that poultry litter out onto their fields. And they were getting soil test levels for phosphorus that were like 300 parts per million, which is pretty high. And when that soil and that phosphorus got into local waterways, they were seeing algal blooms that were decreasing water quality and actually were causing problems for cities that were now having to clean that before they could distribute that water for different areas. So if you're going to apply a organic fertilizer, particularly manures, this is particularly true for manures, you also want to be aware of the other nutrients that are a component of that as well, because the microbes that will help build up in the soil, the microbes will be breaking that down and releasing those as well. And now phosphorus is not mobile in the soil. So you're going to lose that if you're losing soil into that, or the manure itself. Let's say you miscalculate and you put out the manure and then it rains like the next day. We actually had a producer who was doing that, and we were collecting runoff water from their field, and we had these flumes, and we went and collected it, and the flume was basically full of all of his manure that he had put out on his grasses. So you want to also, like anything, keep an eye on what the weather's doing, and hope, this is probably not the best weather. It's great for getting into the field, but it's not really doing much for growth for anything right now, because I think we're over 30 something days without any rainfall. So things to keep in mind. Now, that's not necessarily true for things like cover crop material. There will be the nutrients, the phosphorus and that kind of stuff with it, but the levels aren't quite as high. And so they don't generally cause the same problems that we see with things like manures.
So like whenever we're applying manures, I have a large pile that we bought an old horse training facility. So we have this huge 3 large piles of horse poop. And so whenever we want to spread that or whatever, are we looking for a day that it's rained before and it's not going to pour? How long to affect our ryegrass that we just planted? When should we apply that?
So it's going to take some time for that to break down. I would say, as far as soil conditions go, if you can wait till a little bit after a rainfall so that things are actually starting to grow and reproduce, that would be great. If you can do it in Louisiana before it rains, for a couple of days and give it a chance to sort of start incorporating, that is beneficial. But yeah, I mean, that's the fun part about working in Louisiana is it looks fine and then it comes up and pours for about 30 minutes, and which wasn't even in the forecast. So, nothing's going to be perfect for that, but it is something that you might want to put out, like so inorganic fertilizers, depending on the form that you put it out. If you put out like a urea or something, that's going to convert to plant available forms fairly quickly, within, a week or two. For organic fertilizers, it's going to take longer. And I don't have an exact date for that, like, how many days or months, but it is going to take more time for that biomass and that material to be broken down by the microbial community and those nutrients to be released. And like I said, you're not going to get the same amount out of it. So, giving yourself enough time to let those biological processes happen is your best bet.
Sure. Does anyone have questions besides me?
Yes, if I could. I don't know if you can hear me or not.
Yeah.
Yeah.
I'm Caleb Alexander. I'm out of Livingston Parish area, and my wife and I just bought 39 acres up in Killian, Louisiana, and one of the things that we're interested in is what your recommendations are for getting just initial tests on our soil. What's important to test, track it, and because a lot of these practices we're excited about implementing over the course of our lifetime. What's the best way to go about and tracking that?
So as far as testing goes, the basics, like with nutrient testing, are going to be, I would think, the most beneficial for you, because it's going to tell you whether or not you're going to have deficiencies, things that you need to address. Soil organic matter or soil organic carbon are very important. What I will say on all of those is be aware of what test you're running and what lab, or when you send it to a lab, what tests they're running, because some labs will do different methods for measuring the things. So for like organic matter, we have a combustion method where they will basically put it in a furnace and burn off all the material at about 450 degrees. Some labs will do a chemical reaction, and so that's going to impact the data that you get back. And so I've had situations where a farmer was very concerned because they were getting very different numbers than what they'd seen before. And it came down to, we were using different labs that used different processes. So when you get your data done, maybe just keep track of like, what method are they doing? That's the same thing. So nitrogen's pretty standard. Phosphorus, there are a couple of different processes that is used. So mainly my recommendation with that is just be aware of what tests they're doing. So when you get your data back, how they kind of relate to each other. And if there's a difference, is it a lab difference or is it something else? From a soil health perspective, things like soil organic carbon are something that I would suggest tracking over time. I don't know that I would suggest doing it fairly often because it can vary over time throughout the year. But doing it around the same time each year is also beneficial. There's a lot of soil tests out there that you can look into doing that, you know, it's something that if you are implementing a new practice, it might be good to track something like soil respiration. The caveat to soil respiration is it's not a great one on its own because it tells you that your microbial community is active, but it doesn't necessarily tell you what they're doing. So it's a good one to see that if you're seeing changes in your microbial community. But again, most of these are ones that I would suggest tracking maybe once a year, not even that often for some of these practices, because it is going to take time to start to see changes and see benefits. Some labs will do something called aggregate stability. If you're going to be implementing practices like changing to no-till or something, or incorporating organic material, that's something that I would look at every few years to see change. I definitely wouldn't do that one every year because you're going to see variation and it's not going to be as useful from year to year, but it's something that maybe every couple of years or every 3 or 4 years would be useful to see, are you having the desired impacts and changes? And then I would love to say like some of the biological assessments, but some of the things we're seeing out of those is that they are how useful they are at being able to distinguish between changes. And so there's labs like that are available. Unfortunately, there's not any really in our region at the moment. So if you send them off to another lab, so like Ward Lab will do the Haney test, which could be a beneficial one. There's a lab at Cornell that does soil health assessments. On any of those, the data that you get back is very often based on samples from the region that lab exists in. So like Cornell's, it's getting much better, but a lot of their data initially was from the Northeast or the US, which doesn't necessarily reflect what we have going on here. So that's another thing. If you're looking for soil health, try to find a lab that is closer to you, that maybe has gotten data and built up a database that is more similar to your climate conditions and that kind of thing. There is a whole suite of analysis that you can do. Things like pH and EC are very beneficial. Soil nutrient testing, I think those are all things that I would recommend looking at on a fairly regular basis. As far as some of the soil health assessments, things like respiration, aggregate stability, and those kinds of things, those are going to be where you've implemented new practices and maybe something that you do on a less frequent basis. Soil organic matter, I think would be a good one to do, but again, try to do that at the same time of year each year, so that you're getting a representative sample of sort of what's going on in your practice. So, you know, if you were in a row crop production, maybe do it at the beginning before you plant or after harvest, and do it at that same time. So you're getting the same sort of processes in before you take that sample each time. As far as sampling the field, you can that's going to depend on, you know, how much you want to invest into sampling. You could do grid sampling where you break it up into 3 to 5 acre areas, and then take multiple samples in that 3 to 5 acres, combine them and send them in. But that's going to be a lot of samples for one area, even on, you know, I believe you said 30 something acres, or maybe it was 60 something acres.
39.
39. So, you know, you could do that. But that can, especially if you're doing the soil health assessments, that can get really expensive. So what I would suggest is look at your field, and if it's got any areas that are kind of uniform to each other, sample those and sort of break it out into different areas. And, you know, maybe you have a low spot so that you can make recommendations across the area that are specific to, or maybe specific to that area of the field, because We have a 100-acre field that we're working on for this, and we can see that in one area we have a phosphorus deficiency that's probably about 20 acres of the entire field. And when we look at the yield data, it's not necessarily showing up anywhere except for that one corner. So that allows us to treat that one area of the field and maybe give it a little more attention, and we're not applying that wholesale to the entire area. And so you could potentially take that 39 acres and break it into smaller sections. And that gives you some ability to do a little more precise measurements throughout the area. And then you can kind of decide on like how many samples you want to pay for to determine how many sample, like how to break up that field as well. 0 to 6 inches or 0 to 12 are the most common, depending on what you're going to sample or what you're going to plant.
And I wanted to add, Caleb, that Southern University actually will do that as a service.
Mhm.
I don't know if LSU has that or not, but Southern will come out and actually pull your soil samples for you.
Awesome.
I
won't
don't,
do-
and
Yeah.
I'm
Sorry,
sorry,
go ahead.
Lisa.
No, go ahead.
I don't know. I know you mentioned 2 labs. Can you tell us any other labs that are doing soil carbon and soil organic matter that might be closest to us?
So for soil organic matter and soil carbon, you can, like a soil testing lab here at LSU will do those. For the more specific assessments, if you're interested in things like soil respiration, like enzyme activities or some of those other recommended soil health assessments, you're going to have to go to something like Cornell or potentially Ward Lab for some of the sort of NRCS recommended soil health assessments. And those would be fine. It does get expensive. And some of the data some of their recommendations are based more on Northeast and Northern climates. So it might be better just as a sort of tracking mechanism and not necessarily for recommendation purposes.
Sounds great. What other questions do we have? That was a great one, Caleb.
I think I talked too much.
No, you're great. There's so much that we don't understand going on under the soil. And this was, I'm sure, an extremely basic thing for your mind, Lisa, but it's a lot, for us as producers, we don't always understand even a fraction of what you presented today. So this is wonderful information for us to be able to refer to.
About entire semester. Well, we go into a little more detail on certain processes, but this is about a semester's worth of topics. I'm actually teaching a soil microbiology course this semester with labs, so you also don't get that to the hands-on experience that way either. So, but yeah, I can talk about this all day, so I tend to ramble. So I apologize.
Lisa, are you open to questions later on after this webinar?
Absolutely.
Is over,
my email address is there, my office, actually, yeah, that's my office number. And then email is probably the easiest way to get a hold of me. Just because between field work and teaching and everything, I'm not necessarily in the office that frequently. But yeah, absolutely. Anytime if you have questions, I'm happy to answer them.
And that ad, is that your Instagram or Twitter?
That is Twitter.
Okay.
Which is not extremely active. We are actually trying to hire a communications person to help us be improve our social media presence with a couple projects that I'm on, so that all the data and everything that we're collecting, we can actually share and make it more available to people.
Awesome.
Also,
Is it time?
Any data that you want shared, we would be happy to share it on our social media accounts as well.
Fantastic.
So we can talk about it.
Yeah,
definitely.
That would be awesome.
But yeah, if we don't have any other questions, or if y'all are thinking of your questions, we're going to do the t-shirt drawing really quickly. So Lisa, do you mind taking your share screen off?
Yeah,
and then
no problem.
Bethany is going to do our magic wheel.
Which I saw this t-shirt on the website the other day. Those are pretty awesome t-shirts.
They're really great and super soft.
They were designed by a girl that works at Twyla This Week in Louisiana Agriculture.
Okay, can y'all see it?
Looks good.
Okay.
Nice. Well, congrats,
Congratulations.
Caleb.
Oh, thank you. That's exciting.
I will send you a direct message, and if you don't mind replying with your address and your t-shirt size, and then we'll ship that to you this week.
Absolutely. And I just want to say thank y'all so much for putting this on. Lisa, that was great. And I hope y'all continue to try to do more and more of these. It was awesome.
Thank you. And like I said at the beginning, this will be recorded, so we'll put it on our YouTube page today and email a link to everyone that registered. So if you want to go back and read through or listen to all of the information, this was an awesome presentation, especially for someone that isn't a science background. I really appreciated how approachable it was, and I took a ton out of it. So definitely feel free to go back and also share with other folks that might be interested. And we'll stay on if anyone has a couple other questions, but if not, thank you so much for joining us today.
Yes. Lisa, I did have another question. Rickson, did you have a question? I know you're unmuted. Maybe not. Can you hear me okay?
Yeah.
Lisa, I was wondering if one wants to start a no fertilizer system. I know this is something that we have struggled with in our farm. We really have this strong desire, I'm sure everybody does now, to go as a no fertilizer system. But in our first year of doing that, after this was land that has been overgrazed on one of our leases, very compacted soil. I mean, we're probably talking about two different problems here, but where in the world can you start? I know you said that the first year, the organic matter that's building is using up part of your nitrogen.
Right.
So, you know, after trying it, we really found that we had to kind of wean the soils from having nitrogen applied every year.
Yeah, and that's something that
How
is
long are
going
we
to
talking
be
about here?
So on average, somewhere between 3 to 5 years is where we start to see that transition. You've sort of completed that transition, and we start to see yields rebound and improve. And if you're able to maintain it, we've actually seen improved yields from what people had seen before as late as 7 to 9 years. So it is something that's going to take time. And in the investment of putting in those cover crops or things that will bring those nutrients into the system without having to supplement them with, an inorganic fertilizer source. The other thing is because you mentioned compaction as a potential issue as well, allowing times for those roots to really get in and start working with that material. It's going to take time for them to break through that compacted layer. They will do it, but it will take time to start help breaking that material apart and creating sort of that cookie come from texture that we like to see at the soil surface. And so that, with perennial grasses, you have the benefit of they will keep putting on those roots as long as the plant is growing. And so trying to have sort of a living plant material. So going from summer grasses into winter grasses, winter covers, legumes, that kind of thing will help to sort of continuously have that process going. And that's one benefit to living in Louisiana, is that you have the potential for 365 days of growth. So, and as long as we don't get like 32-degree weather like we got like a year ago, those roots, even if they're growing slowly, will continue to grow and help break that material.
That's wonderful. And again, if anyone needs to jump off of the webinar, we are finished with it. We're just adding extra nerdy questions. So as far as there's a lot of talk, and in 2 weeks we'll be talking about how and when to graze your winter forages. Are you take the top half and leave the bottom half within that realm, or are you within a grazing 1/3 and leaving 2/3? And are you on the mob grazing side where you compact your herd as much as possible? Where do you stand with that?
So some of the best results that I have seen are sort of that take half, leave half. You know, the more biomass you can leave, the better, because you have that material to sort of grow back. But the other part of it is that you want those animals to be in that field long enough that they are defecating and urinating back into the field. So they're taking those nutrients that they took off the grass that they were eating are getting recycled back into that system. And so we've also seen pretty good results with the mob grazing, where you are giving that time, that grass, that time to recuperate and sort of grow up, reestablish that biomass, also get the roots growing and reproducing. And so being able to move them through the field. I've seen farmers that will do it. The one that I was mentioning, I think he does it every 12 hours. It's pretty frequent. I've worked with a couple that'll do every couple of days. It kind of just depends on, you know, how large your areas are and how quickly they will graze through that. We worked with a producer who was doing it in between sugarcane harvest. And so he had a farmer next door neighbors on his fields that, you know, they would plow out the cane and allow him to come in and plant ryegrass. And then he would use that as a forage early in the year and do not as frequent movement through the pins and everything, but actually saw some improvements in organic material and everything in those fields, because you do have that sort of incorporation with the cattle as they're sort of moving around. They're also helping to give a little bit of disturbance and incorporation, as well as, you know, defecating and applying those nutrients essentially back into the profile.
Since you are the cover crop expert, do you recommend a mixture or a monoculture, or have you found there's a magic number there of number of species to plant in a cover crop system, particularly winter forages?
Right. Most of what I've seen is somewhere around 4 species tends to be a good point. A lot of the research I've seen have not shown a tremendous amount of benefit going higher than that. That's not to say you shouldn't do it, but then you also have to consider cost that's associated with it, and whether you're still getting the benefits when you are planting at lower seeding rates that you get in those mixes for how much you might be investing into them. So one of the benefits of having a mix is that you are getting this diversity of different types of things. And maybe this year's not a great year for cereal rye, but oats do well. And so if you've got a couple of different options, you have things that are also establishing at different times, which could be beneficial. It could also be detrimental, because if something gets established before, say, your legumes, they might outcompete your legumes and you don't get as good a stand of those if the legumes are what you're after. So establishing at different times means that you sort of have this continuous coverage, but you could also outcompete some of the species that you mix. And that's another reason why I don't tend to recommend the really diverse mixtures. I saw one the other day, a farmer on one of my Facebook groups was putting out a 26 species mix. And I was like, that's impressive. I don't normally see something that's that diverse. And it does come back to also how long you're going to have those covers in the field. If they're going to be in there for longer periods of time, then you have time to cycle through those different species. If it's in something like a row crop system where you might have 4 months, you know, we've seen the best turnover of nutrients and nutrient availability in like single species legume mixes. So it depends on what your goal is. If all you want is a nitrogen source, plant legumes. If you are wanting it for weed suppression, throw some grasses in. You know, if you're wanting the bigger root systems or for grazing, mixes are good because you do get sort of that diversity of material that's available. But yeah, like I said, I haven't seen the cumulative benefits of the very diverse mixes, the high number of species, so we haven't really gone that high. I think the highest one we have is about 5 species in it for ones that we're trying, just to see kind of what we get and how much biomass we can produce for those. But yeah, it's sometimes they get really expensive. And if you start putting out things like sun hemp is extremely expensive, and costs for all of those covers have gone up in the last few years. So you kind of have to balance what are my end goals and for that cover crop versus how much I'm willing to invest in putting it out there.
And where are your trials? That might be a really neat thing for us to come see sometime as a
Yeah,
group.
and we actually do try to do field days routinely. A lot of my work is in Northeast Louisiana at like the Northeast Research Station, and the long-term one that I was showing some of the data for, that's at the Winsboro Research Station, back in the back. And yeah, it's been in there for 9 years. We have some that have been in no-till and until cover crops for 8 years at the Northeast Station. I do some work with like Brenda Tubana, and sugarcane here in the Baton Rouge area. I don't really have anything in central Louisiana anymore. We used to do a few trials there as well. And our on-farm trials, most of those are in Northeast Louisiana as well at this point.